How many are familiar, at least know the basics about 1,000 Cankers disease? Okay, some of you don't, so I will be starting with some of the basics. Good timing. I think last night, copies, Beth was telling me the story, last night, copies of this new brochure on 1,000 Cankers disease put out by the Morton Arboretum were just printed, the first printed copies, so I think they're out on the table, aren't they? So pick up a copy of that and then you won't have to take any notes. I am a research scientist, so you're going to see some graphs, and I'll be talking about some of my research, which will give you an update and my perspective on 1,000 Cankers right now in the eastern U.S. So I think you'll be interested in what I have to say, but I'll go over the basics first. Let me get everything I need. So this is an emerging problem, and it's, does anybody know where, well, let's just start here. We just had lunch, right? So the alphabet soup, we've heard that all morning. We heard about Dutch elm disease. We heard ALB, Asian longhorn beetle, mentioned. We heard SOD. We've heard emerald ash borer we know about. What's BBD? Beech bark disease. I'm a pathologist, so you know it's got to be a disease, right? Beech bark disease, and now we have TCD. So we have a plethora of acronyms and pest and disease problems. So I'm going to start with the basics. What is TCD, for those of you who aren't familiar with it, and why are we concerned about it? So it's a complex, it's an interaction between an insect, a very tiny insect, that's a bark beetle, Pityopteris juglandis, a walnut twig beetle, which now is a misnomer because it attacks more than just twigs, and a fungus, Geosmithy of Morbidax. And when these two get together on a susceptible host, and these are juglans species for the most part, but in the eastern United States, it's Juglans nigra we're most concerned about. We have this equation for what we thought was disaster, walnut twig beetle plus this fungus, plus a susceptible host, and we get this terrible new disease. This is a tiny beetle, and I have, thanks to Stephanie, I have some beetles in a vial I can pass around. I had brought, because I'm on the road, I had just brought Riker mounts, but it's a tiny beetle, 1.8 millimeters long, very tiny. And you'll see as we pass these around, this is just to orient you. I've got two Riker mounts, and one of them has a vial with the beetles. And again, thanks to Stephanie, she has a fancy hand lens that has a light on it. So those of you who get this one, you can turn this on and actually look for these tiny beetles in the Riker mount. And you can just see how tiny they are. So why don't we start it, and you can pass it on your own from table to table. But it is a tiny beetle. And here's a small flashlight with another Riker mount and another hand lens. These are galleries. These are not really, it's not a natural situation. I was working with a research trial, and we could slip the bark off and see the galleries of this beetle. But at least you get to see the beetle in size relationship to its gallery. So that's why I like it. Anyway, so it's a tiny beetle. But guess what? The good news, I guess, is it's not from Asia. All right. So it's not even an exotic beetle to the U.S. It's a native to the U.S., being found in Arizona and New Mexico, and we think also it's further down in Mexico, and we think its native host is Arizona black walnut. And very tiny. Pencil point and the insect, and a tiny, tiny hole. And if you know walnut, especially eastern black walnut, if it's a larger diameter branch or a bigger tree, you have furrows, so it's even harder to see the damage. But tiny holes, tiny galleries, and this is when you're going from the outside of the bark in. You carefully scrape away with a draw knife or, say, a buck knife, and you see these very tiny galleries. So it's very hard to, you kind of have to calibrate yourself after looking at something like a cerambycid that's so huge. And this fungus species, Geosmithy of Morbidax, is a newly discovered species, first described and published 2010. Geosmithy of Morbidax, it is among a number of other species of Geosmithia that are not pathogenic to plants. And in fact, Geosmithia species up till 2010 were insect associates, very commonly associated with bark beetles, especially in Europe, where they were first intensely studied, and now, of course, we're looking at them closely here. Now we know they also occur on ambrosia beetles as well, and actually a weevil, which we found a couple of years ago. So this is the first species, though, that was known to be a plant pathogen, and it's on the juglans. It causes small cankers, and I'm just going to be talking primarily about eastern black walnut because that's what we have here. It does, by the way, attack the insect and the fungus, can cause holes and then cankers on butternut, but butternut, as you know, is endangered by butternut canker disease. So that's another topic for a future meeting, I think. But anyway, this causes bark cankers, and just to refresh your memory, this is the outer bark here, this is the inner bark, and here's the vascular cambium and the wood and a cross-section of a typical woody plant, and these cankers are in that inner bark. So when you're looking for them, you very carefully slice with like a draw knife or a fairly large bladed knife, and you look for these. I want you to take a close look at this because I'll be talking about other cankers later on. This is about the size of a quarter. So these are very tiny cankers. This is a typical Geosmithia canker. And you can see, again, this is a needle probe I used in the lab, so they're not very big. So we have a tiny insect, a pretty small canker. But thousands of cankers, of these fungus cankers, resulting from even more numerous attacks by the walnut twig beetle, are formed, and they coalesce and lead to branch death and then whole tree mortality. So you can see how they've coalesced here. These are actually pictures taken in Colorado when we first went to look at this disease in Colorado, where it was first described, the pathogen and the insect and the disease itself. But a tree would die very quickly. And when a tree is succumbing to this disease, you would find epicormic sprouts that could occur. And as you know, I've described, the cankers are in the inner bark, and what happens is that eventually that stem is girdled, and when the phloem or the inner bark is disrupted, you're not getting the food that's produced, of course, in the crown of the trees through photosynthesis. It doesn't get down to the roots, and so it's slow starvation is what's happening. It's not like Dutch elm disease, where the fungus is plugging the vascular system and the tree dies more quickly from lack of water. This is lack of nutrients. So there's a difference here. Symptoms develop more slowly. They develop slowly as more walnut twig beetles attack a tree, and then more cankers form. But what happens is the insect will come in, and this was the story in Colorado. A tree could have been attacked for, many years. 7 to 20 years is what the person who discovered the insect in Colorado, Whitney Cranshaw, said it could be 7 to 20 years that this insect could be coming into a tree, colonizing it, and you don't see it because it's so small. And the crown looks normal. But then once enough damage has been done and enough cankers are formed, you start to see symptoms such as these flagging crowns. And you're used to this with Dutch elm disease, so this isn't unusual to you, I'm sure. And then there can be some discoloration that may look more bronzing, or it can be yellow or just brown leaves. And then a wilting appearance. So that's pretty classic for thousand cankers disease symptoms on a walnut. What happened, we think, is that the walnut twig beetle jumped host. I told you it was native in the southwestern United States on the Arizona black walnut. And you know how people carry plants every which way? Well, back in the 1800s, even early pioneers carried eastern black walnut to the west, to Oregon, on the Oregon Trail. And so eastern black walnut's been planted in the west where it's not native for, over a century, almost 2 centuries now. So it's distributed in the west western U.S., but this is the native range of eastern black walnut. And, butternut, Juglans cinerea, is in the eastern U.S. as well. And then there are the western species, Juglans major being the Arizona black walnut, California black walnut, and Juglans heinzii. And then there's a bridge species, Juglans microcarpa, that occurs in Texas that we're concerned about. We were concerned about it as a bridge species. But these are the hosts, and this is in order of susceptibility of these trees to colonization by the walnut twig beetle and canker formation and branch dieback and death. So eastern black walnut is highly susceptible, as is butternut. And then we get the hybrids with Niagara and other hybrids that occur. And then as we go down, little walnut, the microcarpa and the Arizona black walnut are the most resistant to this disease complex. So now hopefully everybody's caught up. What is TCD, right? If you didn't know anything about it before. So why are we concerned? I'm just going to give you 3 reasons. There are more than this, but first, eastern black walnut is widely distributed in the eastern USA, and this is a distribution map of black walnut in the eastern US produced by the U.S. Forest Service, Forest Inventory and Analysis, and just look at the green and you know that's where eastern black walnut occurs. So it's widely distributed in our forests, but we also know it's an urban tree too. It's kind of a messy urban tree, but it's an urban tree, and some people like it in the urban environment as well. But secondly, eastern black walnut is a very valuable timber species. It's probably the most valuable tree species that we have for export, and even timber uses within the U.S. But we export a lot of, or we used to anyway, black walnut until this problem has kind of slowed down our exports. But it's used, as you know, for a lot of different products. The nuts are also a commercial product as well. Eastern black walnut is another industry that's affected by this disease. And another reason to be concerned is now we're having a commercial trade within the U.S. as well as exportation of black walnut to other countries is being hindered because of this disease. It is not an exotic disease, as I said. The insect's native. We think the fungus is probably native to the U.S. So APHIS, USDA APHIS, is not regulating this problem. But the states have decided to do that on their own. And so we have a number of states. I'm not even sure how many states have quarantines now, probably about 16. But so there are state quarantines that prohibit the movement. They have various clauses in the regulations, but it prohibits the movement of different, types of, whether it's walnut logs, any walnut material, or walnut slabs, things like that, or burls, from states with 1,000 cankers disease to states that don't have it, pretty much. And then within a state that has it, a lot of states do have decided to implement county quarantines, not statewide quarantines within their own state. So it really has caused problems in terms of commercial trade. So those are 3 reasons why we're concerned. All right, where does it occur then? It's in the U.S., but a little history lesson. The insect was first described in New Mexico in 1928, and I think there were insect museum specimens of the insect in California, I think the late 1950s, I believe. So the insect has been documented in quite a while ago. In the early 1990s, reports of walnut decline and mortality were documented in Oregon and Utah. How many know about decline diseases? You've heard of different types of decline diseases, oak decline. Well, it's pretty nebulous, isn't it, to say something has a decline disease. It doesn't tell you about what's causing it, does it? Well, this was one of those, walnut decline. So it was reported, but at that time nobody was really looking at what insects or pathogens might be involved. Then in 2001, there was a report of black walnut mortality in northern New Mexico, and the walnut twig beetle was associated with that Juglans nigra. It was Juglans nigra in New Mexico, and remember, it's out of its native range. And they found this walnut twig beetle not just attacking twigs, but larger branches in the main stem. So this is where we think it's jumped host. We think it jumped from the Arizona black walnut, which does occur in New Mexico as well as Arizona, and jumped to this highly susceptible species that was planted. And then in 2003, Ned Tisserat, a pathology colleague, and Whitney Cranshaw, an entomologist at Colorado State University, got reports from actually a city forester in the city of Boulder, Colorado, that trees were dying and you better look at this. And so they worked with that city forester, and by 2007, they realized it wasn't just drought. They just thought it was drought damage, and walnut was suffering because it, really wasn't meant to be in Colorado. And so it was by 2007 they had discovered the walnut twig beetle was present in great numbers, and they found these cankers. And after I think about a year, Ned Tisserat finally decided Geosmithia, this new Geosmithia species, was involved, and it's carried by the insect. So it's a recently described problem. So it was in 2009 the first description of this disease was published. So it's a really new disease. So this is the distribution of the disease as of this month, actually. And what happened after the first description, of course, there was a lot of concern. We want to put a barrier right here, right? We wanted to erect a wall so the beetles wouldn't fly east. Or we were concerned about the Juglans microcarpa in Texas serving as a bridge for the insect and for the fungus to make it across to the highly valuable eastern black walnut in the east. Well, we didn't have to wait long, and all of a sudden we found the disease was found in Knoxville, Tennessee, and then in Richmond, Virginia, and right along the Great Smoky Mountains National Park here, and then subsequently in Doylestown, Pennsylvania, and it's been found in Maryland now, and southwestern Ohio, just north of Cincinnati. And this spring we had the first report of the walnut twig beetle in Indiana, 14 miles from the Ohio border. But so it is in the east. How did it get there? Well, we know in Doylestown, Pennsylvania, it was an urban or suburban setting. It was actually I hate to say this, a tree care worker who did woodworking as well as a hobby, and his trees started dying, and he reported it to the Pennsylvania Department of Ag, eventually found out what it was. Well, he had been ordering walnut burls, I believe, or it could have been slabs with bark, I can't remember which, but from California, where TCD is native. So these are all, they're not so concerned about thousand cankers disease in the West. But so he was importing this stuff for his woodworking hobby, and we think that's how it got established in Pennsylvania, and could likely have happened in the other states, or at least Tennessee and Virginia as well. In Ohio, the walnut twig beetle was first found in a mill site, where it's a bulky mill, actually imports walnut with bark attached at that time to make specialty things like wood dashboards for high-end, luxury cars. So we think that's how the walnut twig beetle got into Ohio. So you can see now we do have it in the eastern U.S. and primarily in urban areas, but it's gotten into the forested areas that are on the edge of the urban areas. There are annual surveys that have gone on since actually, I think 2010 is when we started in the eastern U.S. And this was actually, we started surveys before the first find in the eastern U.S. And they were visual surveys. Well, since then, an entomologist, Steve Seibold, was able to determine that there's a sex pheromone associated with the walnut twig beetle, and so then they were able to synthesize that, and commercially now we have a lure available that's used to trap walnut twig beetles. So our survey has gone beyond just visual surveys. Now we have you know, the Lindgren funnel traps. And we just use 4 traps in, you know, as stack traps with the lure. And so every year, I don't have the latest results, but every year USDA, APHIS, and the U.S. Forest Service fund these surveys, and some states fund their own additional surveys as well. But they're trying to every year update, just like EAB and other major pests, where the insect has been trapped. But all the green that you see here, the green dots are no walnut twig beetle, and the black dots are no walnut twig beetle, with the green being Forest Service surveys, funded surveys, and the black being APHIS. And then the yellow are counties with 1,000 cankers disease, and I've already told you about those areas. You can't see it in some of these states, but anyway, so there is an annual survey going on. This is the trap I described to you verbally. So 4 funnels is all you really need. You can use the longer ones, but this will suffice. Put on a pole about 20 feet from a walnut tree, especially if you got a walnut that looks kind of like it's declining. So this is a standard trap that's used with that lure. And the lure looks like this. I don't know if you can see it. The commercially available lure looks like that. And you just, in this case, we stapled it to a walnut branch. So what happened was, I was involved in the state of Maryland with trying to, they had trapped walnut twig beetle using these traps in 2013, I think it was 2013. They said, can you help us isolate Geosmithy of Morbidax? I'm a pathologist. And so I said, yeah. So they were sending me branch samples during the winter. Of course, they couldn't see any symptomatic crown. You know, they were just going out and, well, I think I see some holes here. We'll send this to her. Well, I got kind of tired of that. So after 2 tries of getting branch segments in the winter, I said, just wait till summer. But in the meantime, Steve Seibolt in California, who developed the lure, they have walnut twig beetle all around his laboratory. So he said, well, why don't we just take a branch segment, put the lure on it, and just hang it in areas where we've got the traps. The problem is when we trap the insects with this, they go into a liquid, a collection liquid, RV antifreeze. And it's just really hard to isolate the fungus from those beetles. So, you know, Maryland had a find of the walnut twig beetle, but they didn't have a find of Geosmithy of morbida, and so they weren't going to tell the world that they had TCD until they had both pieces. So then they put out this branch type of bait, and then they sent me these in August of 2014, and I isolated the fungus for them. So now Maryland officially has TCD. We have national survey guidelines, so hopefully there's some kind of consistency across the surveys that are conducted in the eastern U.S. And if you're interested, this is a great website, not just for the survey information, but all kinds of information about 1,000 cankers disease. This is actually on the APHIS website, but I think you would benefit from knowing this address more. And there's a link to the survey guidelines. Lots of other information at that address, by the way. So, okay, and I came yesterday afternoon so I'd have a chance to talk to Frederick Miller, who works here at the Arboretum, because I wanted to find out more about the surveys going on here. And I think Kelly, who just talked this morning, and Scott, you can help me here, but this is based on what Frederick had told me yesterday afternoon. But he said USDA APHIS and CAPS surveys go on for the walnut twig beetle and TCD. I'm sure you do more than ports of entry wood product sites. You probably do transportation corridors, but you coordinate. I know you coordinate with each other, and then you coordinate with Frederick. That's what he's told me. And so that you're covering the state of Illinois in what seems the most logical places. So Frederick is actually working, focused on state lands and county lands. And in fact, he left at 6 o'clock yesterday evening to go downstate to put out his traps for walnut twig beetle. So that I just thought you'd be interested in knowing what's going on in the state. And this is the link on the 1,000 Cankers website. This is the information for Illinois. If you're interested, Illinois does have a quarantine in place. If you're going to move walnut, you need to have a compliance agreement. and details there. And then there's some more fact sheets and things. So lots of information available for you online for Illinois. All right, now I'm going to get into more of the science part of it and the work that we've been doing. But I'm going to start off talking about work that some other folks have doing that I think that I built my work on. So what do we know about the behavior of this new emerging disease in the eastern United States? We know it was more like a decline disease in the west. What's going on in the east, where we're most concerned about the impact on this very valuable timber and nut species? Well, when I first saw TCD, it was in Wheat Ridge, Colorado, 2010. Those trees, you'd see flagging on a tree, say in June of one year, and the next year the tree would be dead. That's what was going on there. This is in Maryville, Tennessee, which is just a suburb of Knoxville, and it looks very different, doesn't it? So I wasn't the only one asking this question. A number of other colleagues were saying, well, what's going on in the eastern U.S.? Is it really the same as what's going on in the west? Gary Griffin is a forest pathologist, actually retired, but still active, working at Virginia Tech, and I think it was great. Right away when TCD was detected in Richmond, Virginia, he jumped in and said, well, I'm going to start following the symptom progression in TCD trees in Richmond, Virginia area and in Knoxville, Tennessee. And so he categorized the progression. He came up with 4 types. And this was published in a journal, scientific journal, Forest Pathology, just last fall. So he has 4 types of symptom progression. Slowed and quiescent dieback, kind of sounds like a decline, doesn't it? Green foliage wilt with dead clinging leaves, upper tree crown with yellowing leaves and leaflet drop, and then recovery. So he had these 4 types that he documented in those 2 areas in the eastern U.S. And type 2 and 3 were common with what was observed in Colorado, but type 1 and 4 had not been reported. And I think the recovery is very exciting. He had beautiful color photos. It's an online journal, so lots of color photos in this publication. But just to show you, here's an example of the recovery type. And this is this the same tree on the left and right. So this tree, I think he had rated as 60% live crown. In 2010, 2013, it was 90%. So this is recovery. So very exciting. He followed 56 trees in Richmond, Virginia, and 56 trees in Knoxville, Tennessee over this time period between 2011 and 2013. So I think that's very exciting. And then he did several things that are published in that paper, but I'm only picking on these 2 areas. First, the symptom progression, and now I want to talk about precipitation patterns that he documented. So he took the NOAA weather data, and he looked at precipitation in Richmond and in Knoxville for the same time that he was following symptom progression. So I've kind of summarized what's in his paper here. So in 2011 and 12 in Richmond, Virginia, precipitation was low, again based on NOAA records, between January 1 and the end of August of both of those years. And as he was documenting the symptom progression over the same time period, he noticed the correlation that new TCD symptom development was occurring. And then in 2013 in the Richmond area, precipitation was higher in that same time period, but in 2013. So it was more normal precipitation pattern. And it correlated with no new symptoms of TCD observed in his survey. And then looking at Knoxville, Tennessee, between 2011 and 2013, there were very high levels of annual precipitation for that region. And then his survey found no new symptoms of TCD, and associated that precipitation pattern was associated with increased growth and recovery of Juglans nigra. I showed you his pictures. So I think that's very exciting. And so I spoke to the Ohio Forest Health meeting last month, and they said, you're giving us hope. So I think there is hope, and I'll go on to give you some more reasons why I think there's hope for eastern black walnut. But let's keep talking about this, building on what Gary Griffin found. What do we know about Juglans nigra? If you're a forester, you know Juglans nigra is a mesic tree species. It needs moisture. It needs moist soils to grow in. In Colorado, it probably wasn't very happy, especially if it was a drought year. It's shade intolerant. It's, as you know, it's widely distributed, as I've shown you. It does best in deep, well-drained soils and moist and fertile soils and about neutral pH. So this is a black walnut plantation. Does anybody deal with plantation growers in here? Or? Okay, you're probably mostly urban. I work a lot with the Walnut Council and a bunch of walnut growers who have their retirement sitting on the stump out in these plantations. I mean, they're very, very concerned. So you can imagine when they put in a plantation, they really baby that thing, and they site their plantations very well. But what do we do in urban areas? We do lots of fun things to our sites. This was you can't hardly see it, but it's a house that's not too old. It's probably about 15, 20 years old that they developed this site. Walnut throughout that backyard. This is in Butler County, Ohio, just north of Cincinnati. And there was fill put in. These were kind of wildland trees, and then they built the house, put in fill, and then, you got to have lawn, right? They put grass, nice sod around those trees. Well, those trees are stressed. And so all these trees have TCD. So, we see this all too often in urban sites that, we have stressed trees because of whatever reason, whether it's excavations, fill, road construction, just general abuse, as well as natural things like, we might have a year of drought. But anyway, the bottom line is altered sites are unfavorable for Juglans nigra growth and vigor. And this is, like I've told you, is primarily where we've seen TCD in the east. So one thing about how TCD behaves in the east, it seems to be related to drought stress, perhaps, moisture deficits, and we know there is recovery. In fact, the large majority of the trees that Griffin documented with the symptom progression, most of those were either slow decline and quiescent, dieback, or recovery. That was a majority. So, I think there's, again, more reason for hope. So now let's look at the question, what do we know about the insect's behavior in the eastern U.S.? I am a pathologist, but I've always, my whole career, I've worked with insect-fungal interactions, and in doing that, I've always worked very closely with entomologists. And so I am, once again, I've worked with an entomologist at University of Tennessee, Knoxville, and now I'm working very closely with Matt Genzel, who's a forest entomologist at Purdue University. And I work with Fred Miller, and I have worked together too here last year. And I think that's very important if you're working with a disease like this. So working in Knoxville, Tennessee, I said, well, what happens if we I wanted to girdle trees. But in Knoxville, they said, no, you're not going to girdle trees because that means the tree's dead. So they would let me girdle branches. So we went right in the suburbs of Knoxville where TCD was common. And on one side of a tree, we would girdle a branch like you see, very crude, but girdled. And then on the other side of the tree, we'd have another branch not girdled. And working with actually an entomologist from Tennessee State University as well. And she documented through flight interception traps, they were just sticky pieces of paper like window traps placed in the crowns of these trees. She documented how many walnut twig beetles were coming in. This is the average cumulative walnut twig beetles trapped on these sticky sheets, kind of clear sheets up in the crown. And 2 years, 2011, 2012, we did this study. The closed circle are the girdled branches, and the open circle are the non-girdled, or the controls, we call them. So you can see this is the number of beetles. This is between late May when we girdled those branches and early September. And it doesn't take a scientist to figure this out, that there's something about that girdling that's attracting the beetles into the crown of those trees. So there's something we've learned about the behavior. So what is it? I'll get to that question in a minute, but the other thing we did with this particular, it was a pretty small study, we then cut these branches off the end of, after 3 months, so early September, and we cut the branch into a number of segments, and we took 2 of the segments and we isolated for fungi, and I'll talk about that when I get to the fungal side of the story, but 8 of the segments we put into these boxes that we adapted for emergence of insects. So then we emerged all the walnut twig beetles from there, and my colleague, entomologist Bill Klingeman at University of Tennessee, emerged those insects, and Bill just wanted to go on and on, so he went for 9 months with those. But look at that. 5,000 from those girdle branches and 25 from the non-girdle. Now, what happened here is the beetles would emerge in those emergence boxes and then they'd recolonize. So that's not a true number in a sense. So we're building up the population, but still, it does give you the idea that we're getting colonization of those girdle branches. So we know the beetles are attracted really basically to a stressed branch, right? And then my colleague at Purdue, Matt Genzel, was interested in what are the volatiles. First, he was looking at what are the volatiles just from different genotypes of black walnut. Now, he works a lot with the folks who have plantations of black walnut, so he has 10 elite genotypes of black walnut at Purdue that he could study. And so they bagged just the branches with the foliage, and then they captured the volatiles, just produced, these were not injured at all. And they captured those volatiles, and then they released it in this fancy glass tube, and then they introduced the walnut twig beetle at the end, and then they watched. It was a grad student, poor grad student, who had to measure the walking response of the walnut twig beetle. I couldn't do it because my eyesight's so poor now in my old age, but a grad student could do it. So there's this, and from this walking response, the student developed an attractivity index. And so this is the student's data. This is the attractivity index. Of course, the higher the walking response, the higher up you'll see this. And these are those elite genotypes by their clone number. And the point is that there's variation. So attractiveness to the walnut twig beetle, to the walnut volatiles, by the walnut twig beetle does vary by genotype. So that's very interesting. But then I said to Matt, well, Matt, look at what we found in Tennessee with these girdle branches. So he had a student then look at girdle branches, capture the volatiles over girdle branches versus the non-girdle branches where we were working. And so these are the girdle branches, the intact or non-girdle branches. And then we just had control trees here. But the difference is, statistically speaking, there is a difference in the attraction of walnut twig beetles. Many more are attracted to the girdle branches. So we know there's something going on. It seems to be that we call them chiromones. They're plant chemicals, plant volatiles that are released that are attracting the walnut twig beetle. So we know there's sex pheromones produced by the insects themselves, but now we know there's some host chemistry involved as well. So Matt's interested in improving the walnut twig beetle lure by adding a component for the host to that commercial lure. So that's what we learned about the behavior of the insect. So then I asked the question, well, what are other insect pests that come into stressed walnut? And in particular, I was interested in other bark beetles besides the walnut twig beetle, interested in ambrosia beetles. And I had seen on some logs that were passing through the rail yard in St. Paul, Minnesota, destined for the West Coast and Korea. This was in 2010. I saw that weevils were colonizing the bark. And I said, we need to be watching for bark colonizing weevils as well. And the Minnesota Department of Ag had to inspect those logs and certify them to move any further. And I was called in. So we had one weevil species we knew we should look for in 2010. So those were the 3 types or taxonomic groups I was interested in. So I started working with a postdoc, a woman who has a PhD from University of Missouri in Columbia, And we decided we were going to use trap trees, since we could work in Missouri and Indiana by girdling whole trees, not just girdling branches. And so she did Missouri, I did Indiana. And this is just the state of Indiana. There's Lake Michigan and Kentucky's down here. So I covered the whole state with 15, I chose 15 study sites. Each site I had 4 trees, and I girdled them. And 12 of those sites were walnut plantations. There was only one urban site, and that was actually an orchard, but it was near a mill. And then we had some wildland sites, just kind of fence rows. But we girdled and we applied herbicide, glyphosate. This was the end of May, first week of June, and then 3 months later, early September, we felled those trees. We took 2 samples out of the main stem, and we took 4 branch samples out of the same trees. We put them in emergence buckets, and over 3 months, people at Purdue, on my behalf, collected the insects, and then a postdoc in Missouri collected hers, and then she identified all of those. And this is what she found. She obviously didn't, she was busy, very busy, because she found over 16,000 insects. And these were just ambrosia beetles, weevils, and other bark beetles. And we were also looking for the walnut twig beetle, and she found no walnut twig beetles were collected, which was good news. But you can see I put the number of species by these taxonomic groups here. So there was more diversity in these, in the ambrosia beetles species in Indiana versus Missouri. And then we had a few more sites, so we had more individuals collected of the ambrosia beetles in Missouri, but still a lot. And relatively few bark beetles, and then the bark colonizing weevils, surprisingly, we found quite a few. But not nearly like the ambrosia beetles. So predominant ambrosia beetles, we found actually over 19 taxonomic species of these 3 groups of insects, but the ambrosia beetles were the most common, and you've probably heard of these, or at least this one, Xylosandrus crassiusculus, is an exotic, and it's becoming a problem, particularly in nurseries. It's already reached, I think this one was introduced in 1972. It's from Asia. And it's causing problems in a wide variety of ornamental and now in walnut plantations. And then Xylobrinus saxesceni was also introduced, I believe, a couple of decades earlier, not causing nearly as much damage. But this looks very, very much like the beetles that you'll see in what I passed around. But the abundance of each of these species did vary by state. So this first insect was more common in Indiana than Missouri, and the second one was most common in Missouri. But anyway, we had really got top 4 ambrosia beetle species. I'm just showing you these 2. But they didn't, it wasn't the same abundance pattern in each state. And then bark beetles, Pitiophthorus laudus was the one that was most commonly found of the few we found, and it was only in Indiana. And you can imagine, because the genus name's the same, it's a close relative of Pitiophthorus juglandus. And then these weevils. This is the weevil species I'd found in logs back in 2010. Logs from Illinois, Indiana, Wisconsin that were being shipped to Korea through St. Paul. And then we found this other weevil. And again, the pattern was different by state. We found this Stenomymus, very common in Indiana, and Hymation arens, this other one was most common in Missouri. So we had that information, and then we also were interested in the fungi they were carrying, but I'm not going to go into that right now. So now we looked at artificially stressed walnut, just to get baseline data on whether their insect pests will come into these stressed trees. Well, what about TCD symptomatic trees? Well, once TCD was found in Butler County, Ohio, I said, oh, good. I don't have to drive. I don't have to go down to Knoxville, Tennessee now. It's a lot closer to go to Ohio. And so we started looking at this question last year. And so in this relatively new housing development area, we were able to work. And we actually had permission to take down 4 trees, 3 trees in one homeowner's backyard, and then his next door neighbor, we had a 4th tree. But you can see the range of symptoms there. This tree actually had leafed out, probably about 40% live crown in May, but when we felled the tree in September, there were just epicormic sprouts on it. So this tree had died from TCD, and these trees had, tree 2, you can see a little bit of dieback, and tree 1 had partial dieback too. Tree 4 was half dead. So we had a range of symptoms in the 4 trees. We felled the trees, we cut the main stem into 16 1-foot-long sections, and then we took 1-foot sections from each of 4 branches, and we wanted to look at the insects coming out of those and also the cankers and fungi from those cankers. So I'm going to talk about the insect part now. So we took 8 stem sections and 8 branch segments, put them in those same type of emergence buckets. And this is the list of what we found. So this is data just from last fall. 143 of these taxonomic group examples. And Pediopterus juglandis, we only got 1. So they had a rather strange phenomenon happen in 2014, even when the Ohio Department of Agriculture was trapping with the lure-baited funnel traps. In 2014, they got very few. The year before, they got thousands. So we don't know if it was a harsh winter between 2013-14. I think they might have trapped them out. They were trapping so many in 2013. But anyway, this just fit with the trapping data the Department of Ag had. We only got 1 insect. But interestingly, in the blue, you've seen these names before. Xylosandrus crassiusculus, Xylobarinus saxessini were common, and the weevil, the one weevil. So those 3 species accounted for 96% of the total insects that came out of those TCD trees. And, of course, being a pathologist, I had to know what fungi they were carrying, but specifically, are they carrying Geosmithy of morbida? And lo and behold, these are the numbers. That's not hashtag. If you're young, you're going to say hashtag assayed. What does that mean? I used to put NO period, and I was told, no, that looks like no assayed. So I said, I'll put hashtag, but then that means something else. I can't win. Anyway, understand that this means number assayed, number with Geosmithy of morbida. But look at this ambrosia beetle, crassiusculus. Over 65% of those beetles gave us Geosmithy of morbida. And then a lower percentage out of the weevil and out of saxessini, but over 20%. So we have other insects that are carrying Geosmithia. And we call it, in scientific terms, we say it's lateral transmission. They've come out of these TCD trees, which Geosmithia morbida is throughout the stem and in the branches. So these other species are probably picking it up casually as they come through the bark. But they're going to go to other trees. So we're getting this canker fungus being spread to other trees by different insects. So, the problem's just growing, not getting better. So this, I haven't even published this yet, so I hope to get just a short disease note out about this, because it kind of changes how we look at this disease. So what's currently known about these insects, though, that I keep highlighting? So Pediopterus juglandis, the walnut twig beetle, we know it's an aggressive invader of walnut. We know it's very much attracted to the host volatiles, especially if the tree has been wounded in some manner. So we know it's an important, and maybe the first colonist on a declining stress tree. Xylosandrus crassiusculus, we know it's an exotic insect to the U.S. as is this one. But this one is attacking, like I said, right now, a wide variety of woody trees, including walnut. It's aggressive. Chris Ranger from Ohio has worked on this quite a bit, and it will attack healthy as well as stressed trees based on Chris's work. Xylobarinus does not seem to be as aggressive, but it's very, very common. It attacks a wide range of host trees, including walnut. And we don't know much about this weevil. So we have a graduate student starting at Purdue in August, and He has a background in entomology, so he's going to be looking at what's going on with these other species in relation to Pediopterus jugulandus. Now, finally, getting to the interesting part, Stephanie's going to give me her attention now, because we're going to talk about pathology. So I was interested in, well, we know, and I know in Indiana, or in Illinois, Botryopsphera dithydia is a canker fungus, and it causes branch dieback in walnut, and that's been well documented in Indiana, Illinois, and Missouri. Does it occur on these TCD trees? We don't know. There are other fungi as well that we know are known pathogens of walnut. So I was interested in that question, what other fungi occur, and in particular, what other putative or real known pathogens occur on TCD trees besides Geosmithia. So I worked first in Knoxville, Tennessee, and these were the same trees we used to emerge walnut twig beetle from those girdle branches. I took 2 segments from each branch, and we carefully stripped the bark, and we looked for damage. And we categorized, I have a master student who categorized that damage. So she had 2 different types of stains that she described, 3 different types of cankers, and then 2 insect-related damage, buprestid-like damage, and Pediopterus jugulandus-like damage, as very fine galleries. This was more like the typical Geosmithia canker. You can't tell it. We had to freeze the samples to ship them to Minnesota, so they look odd. But anyway, so we characterized that there were 7 types of damage, and then the student isolated from the margins of those different types of damage, the stains, the cankers, the insect damage, and these were the woody plant or pathogens of walnut, eastern black walnut, that we found. So Botryopsphera dithydia, which we know causes branch dieback, Fusarium solani, which is kind of a ubiquitous fungus, but we know it causes a branch dieback on eastern walnut in the eastern U.S., and then, of course, Geosmithy morbida. And the point is, you can see all these colors, right, across canker damage and insect damage. So that raised a big flag for me. I don't think it's all Geosmithy morbida. I think there's more to this story than just walnut twig beetle plus Geosmithia equals TCD. So then in Butler County, Ohio, those same trees we took down, Paul got to do this with me. Wave your hand there, Paul. I don't think he can even lift his arm. He stripped so much bark off his stem samples. It took us 2.5 weeks, I think, to do this. But we stripped the bark carefully off of main stem samples and branch samples. But look at this. Look at all those cankers. And, we found cankers down to every 5 square centimeters. I mean, that was how numerous they were. But look, they're still that same kind of quarter size. They're not huge. So you can see why it was first named 1,000 cankers. Well, then we isolated from the margins of those cankers, and we found Geosmithy morbida commonly, 60 to 80% of the cankers we isolated from on the branches as well as main stem, Fusarium solani, and then a different Botryopsphera, which goes by its asexual state, Diplodia seriata. And so this is just recent, what we found last fall. And so my question as a pathologist is, I need to have a race. I want to inoculate branches with all 3 of these and see who wins, in a sense. Basically, I want to compare the pathogenicity of each of these on the same tree. I'll use separate branches. And so that's what Paul and I are going down to Ohio to pick out the trees that we're going to be inoculating with in June and in September. So I'm really excited about this because I think there's much more to this story that we've missed. So this is just to show you the type of data we collected and to further justify my activities for the summer. This is just for the branch cankers. I showed you that branch segment with those numerous cankers. So there's the typical Geosmithia-like canker, and we isolated from 96 of those. And then there were other types of cankers that I described differently. So we isolated from a certain number of those. And then this is the results in terms of fungi found. So numbers of cankers that yielded either Geosmithia morbida by itself, and you can see for the typical Geosmithia cankers, about 1/3 of those yielded just Geosmithia. But Geosmithia plus Fusarium or Bactriosphaeria, the diplodia, about a third almost. So we had Geosmithia by itself, Geosmithia with these, or we had Fusarium solani by itself, or Bactriosphaeria diplodia by itself. Or here's those other two fungi without Geosmithia. So all this just points to the fact that we've got something else going on, that we could have. These cankers may not indeed be caused by all of them by Geosmithia morbida. You heard all morning about insects causing wounds. So if we have the walnut twig beetle or an ambrosia beetle coming into a stressed tree, it creates a wound. Fusarium solani is ubiquitous in the environment, and it can get in through that hole. Bactriosphaeria actually is an endophyte, and it could just come, that stressing by that insect hole could just be enough to shift it into a pathogenic phase. So anyway, that's kind of my evidence for my thinking of what's going on. So as I said, what do we currently know about those insects that I found? Now I'm saying, what do we currently know about these fungi? We know that Geosmithia morbida is a weak pathogen of walnut. When you cause a quarter-sized canker over a growing season, that's not an aggressive pathogen. Bactriosphaeria dithydia that we found in Indiana and Tennessee is a stress-related pathogen of walnut. And this is all documented in the literature, and it survives in a non-parasitic phase as an endophyte and then can break out as a pathogen under the right conditions. This other Bactriosphaeria obtusa is a pathogen on several woody hosts. We don't know that it's a pathogen on walnut, but hopefully we will be addressing that question this summer. And the Fusarium solani, there's been a lot of recent work on this, and it's found to be a species complex, and work done by Matt Casson at West Virginia University has found that there are 3 phylogenetic species that are confined to TCD areas. And then there's a number of other phylogenetic species. That's based on molecular evidence, DNA type of data. And so this story is much more complex, too, than we thought. So we want to inoculate our branches with the phylogenetic species that only occur in TCD areas to see what's going to happen in our race of the fungi. It's like it'll be fun. So in conclusion, I just want to say, I'm going to make 3 points here. And this kind of getting into my perspective. I don't know if my colleagues out west would agree with me, but Fred Miller agrees. We talked about this yesterday, and Matt Casson. And so I have several colleagues in the east who agree with me. First, there appears to be a sequence of insect species colonizing Juglans nigra in the east as TCD symptoms develop. So we know that walnut twig beetle is very aggressive, so it's likely the initial colonizer, but we know it's closely followed by at least 2 ambrosia beetle species and a weevil. So we hope the research that will start this summer by the grad student at Purdue will look at what's the role of these these other insects in development of TCD, as well as in what's their role in dispersing the fungus. Secondly, we hypothesize that other plant pathogenic fungi, besides Geosmithia morbida, are contributing to canker development and branch death on Juglans nigra, trees that are affected by TCD. But their roles relative to Geosmithia morbida need to be investigated, and I told you how I'm going about that. And then thirdly, other insects do carry Geosmithia morbida, especially those emerging from TCD trees. And we need to look at the relative importance of those other insect vectors in relation to Pityopteris juglandis. How important are they? Do the state regulatory agencies need to take into account these other insects? I mean, it really opens up a can of worms. They probably wish I'd never done this work, but it's just, to me, it's really become much more complex of a picture. And so what I think is happening, my perspective is that we probably have more of a, I hate to say it, typical decline disease, where we have a succession of insect pests and probably even simultaneously several different pathogens involved. So it's a complex disease, just became more complex in my opinion. But I believe that it's very much a stress-related disease, and it's not EAB. It's not an EAB situation that if a tree comes down with TCD in the eastern US, I don't think it's not game over. We've seen recovery based on Gary Griffin's data, so I hope I've conveyed that to you. I think there's hope. I think we do need to understand what the contribution is of these other things. So based on all that, I have a colleague in the Forest Service, Manfred Mielke, who's very much interested. He also on the side has a walnut plantation and very interested. He's past president of the Walnut Council, and he wants to develop best management practices. So this is how far, I'll show you what we're thinking about for best management practices. And we've tried to span urban and plantation situations. So that's how I'm going to end here. First, of course, and you've heard this all morning, we need to stop or restrict the movement or importation of, in this case, walnut material from TCD states or counties within those states that have TCD. And that's, as you know, is done by quarantines. And part of that is just making people aware of those regulations. So the state quarantines, I'm just asking that you would become, at least just be aware that you can't just bring in walnut from Indiana now, since they now have walnut twig beetle. And we documented Geosmithy of Morbidax there several years ago on the weevil. You can't just be bringing in walnut from the state next door. Needs to be certified if you do bring it in. Need your help in educating folks, outreach and education, especially to woodworkers. You know, they're looking to California maybe for the highly prized walnut burls, and they want to bring it in for wood turning or something. Well, they may not know about this problem. So if you know someone who's a woodworker, make sure, and if they work with walnut, make sure they know about this problem. They are now fumigating these burls and these walnut slabs with bark attached and shipping them to the east. So they are using methyl bromide to fumigate from those commercial companies. So that's good. But still, people need to be aware. Mill owners, the find of the walnut twig beetle in Indiana, it was trapping done last summer, but they just verified it in February of this year. It was a mill. It was a small mill. And others who import wood. So I don't know what your background, where you work, but we could use your help. And this one, perhaps, people do use walnut for firewood. And don't move firewood. You've heard that over and over and over again. So here's another case for why we don't move firewood. Prevention continued here. In general, I hope I've convinced you that Juglans nigra is a mesic species, and it needs to go on a site that's appropriate for it. You know, I wouldn't replant it in Colorado. So anyway, you wouldn't want to put it in a dry environment or on soils that are well drained and drought prone. Now I'm going to get more specific in plantations. If you know anything about plantation management, one of the things they do, they plant more trees than they need, then they go back in after a number of years and they decide on their crop trees versus their non-crop trees. They want to release their crop trees to get better growth, and the way they do that is they girdle the trees. And so now, after all the studies we've done, we think they can still do that girdling. They girdle the trees so they die slowly, and they don't want to take them down in the summer because they would cause a lot of injury to surrounding their crop trees. So they've been doing that at different times of the year. We're suggesting now they girdle in July and then get those trees out in the winter. Before, if there's walnut twig beetle, if there happened to be walnut twig beetle in there, they get it out before the adults would emerge, or even the ambrosia beetles, Xylosandrus cassiusculus or Xylosandrus germanus. So they would prevent building up pests, insect pests in their plantations that way. And then in urban areas, minimize impacts to trees, you know, whether it's during construction, during road work, during development of a new housing area, water, fertilize, vegetation control. You know, what you know about mulching around trees instead of having turf under there. Early detection, and again, I'm just reiterating what you've heard. I just ask for your support of your state or your agency's effort in walnut twig beetle monitoring and TCD surveys. So I know Fred Miller told me yesterday he really seeks a lot of input from folks as to where he should put out his traps. And then report suspicious trees to appropriate agency or office. And I assume that's the Illinois Department of Agriculture, or is that correct? If you see it, okay. So somebody can follow up on it. And then management and mitigation. So this is assuming you've got walnut twig beetle and geosmithy already established. These are what we're planning to do in Ohio and in the one county in Indiana. Sanitation and treatment of that infested material is imperative. Again, maybe not eradicate if we can't, but to keep the population of the insect near zero and to prevent the fungus from building up. So that means either burying material, burning it, chipping it. Even chipping it can be a problem because that beetle is so tiny. But just proper disposal of it. And then good cultural practices. So minimize host stress where possible. Avoid wounding trees, because we know if we girdle a branch, we're just attracting walnut twig beetle. In plantations, we think if they first find, we haven't yet found TCD in a plantation of walnut, but if it happens, they should of course try to eradicate it from that plantation. But if it gets established, then they should prune hard in those crop trees. Because you just want to prevent the walnut twig beetle. Once it gets into a branch or a main stem, and even if that dies, that beetle, it just has the habit of just reinfesting. But you know what it's doing? It's building up its population. So that's why it's important to get that dead material out of there if you've got walnut twig beetle established in a plantation.